Hello, my name is Karen Pham, and I'm here with the POKY team at the University of Colorado, Denver. Today, I'll be showing you a tutorial on how to submit structured calculation with POKY Builder using simulated data. Before we get started, here's some more background information on POKY Builder. There is a wide array of data inputs for this program, with the necessary inputs including your contact information, chemical sequence, chemical shifts, NOE data, and your structural restraints. You as a user can also add in disulfide bond and system ruling information, adjust tolerances for nosy data, and you also have the option of using the PAXI database to increase the accuracy of your protein structure. There's also different types of calculations and computer servers as well. So a little bit more deeper look into the calculation options. You can start your initial calculations with the Adana DOS automation or the SS Ponderosa X refinement. If you want to use your own constraints, then you can also do refinement with constraints only X. For any final and larger calculation with water solvation, you have the option of using implicit and explicit H2O. So to get started, you're going to be opening the POKY app. Once you do, this main menu will show up with the POKY application. And to get started with simulating our data, we'll be using the 2-character code ME. So it's going to be asking for a BMRB number. Today, we'll be using the example of ubiquitin with a BMRB ID of 6457. Click OK. We're using solution state. And then since we are starting from scratch, we want the backbone, aliphatic sidechain, and 3D nosy information. For the sake of simplicity, we will be using the Norse level of 1. And then we will keep the approximation power value as the same. And now we're going to choose the parent directory. So I already set a directory in my desktop called Ponderosa Trial. Once you click open, the app is going to work on generating all these spectras for you. This process does take some time, so I already went ahead and ran this program for you. So ignoring the other files, it's going to upload a bunch of spectras with the from via .ucsf file. So I'm just go ahead and upload it here for you guys. These spectras are correlated to your backbone aliphatic. But the main focus on today is carbon and nitrogen nosy data. So here's our N nosy and C nosy spectra. And then here's a little bit more example on the other spectra types here. So then the next step is to download our chemical shifts related to our protein. So we can go to the residence tab, and then down here, we can just go ahead and do the download button. This is going to pull from the BMRB database. So we're going to keep the ID as ubiquitin the same. And here is our chemical shifts. So then our next step in this process is going to be assigning and labeling our peaks for our carbon and C nosy. So I'm using the 2-letter code C3, and it's going to be opening the Ponderosa structure calculation. So Ponderosa is a much older version of POKY Builder. And then we start by typing our name. My name is Karen. The email we're going to use, which is going to be Ponderosa example at gmail.com. You're more than welcome to use your own professional and university credentials or your personal email, whichever your preference is. And we're going to name the project as tutorial. Here, we use the select the type of spectrum we want. This one's auto filled in as the N nosy. So now we can select the type as 15 nosy. And here we're going to specify the spectrum we want to use as the carbon nosy. And then we're going to label it as the aliphatic and not the aromatic 13 carbon nosy. And then the very bottom here, we're going to check the box called apply auto peak picking. Next, we're going to run it. This will take a couple seconds, but as you can see here in our N nosy spectra, it's going to zoom in. the peaks are properly labeled. So once you click run, you'll get an email, as you can see here, that says Ponderosa job submitted, along with the correlated job ID. So um, with the with this process also takes a long time. So I already went ahead and ran this program. You'll get a secondary email that either tells you either your Ponderosa job succeeded or failed, along with the same correlating job ID. You can go ahead and click this link. This link will download a zip file for you containing all your data related to the structure calculation. And just click OK. And then I went ahead and unzipped that file into our parent data directory, or just our parent directory. And remember, our job ID ends in 6063. So here is a bunch of statistic files. And then you can just go ahead and click evaluated. This is where you find files related to your restraints, as TBL files, more statistics files, and also PDBs for your preliminary structured data labeled as final or finalx.pdb. So the next step is to refine our preliminary structure. And from there, we'll be using POKY Analyzer. Now I'm going to set up my directory, my working directory, which is going to be the job ID from our preliminary calculations. You can click open. It's going to open a bunch of little windows for Python commands, your chemical shifts, just general database sequences, and your package contents. So here we'll upload your backbone information. And then a little bit more of a POKY Analyzer is a very nifty tool. So you can show your NOE like bar chart. You can also see the Ramachandran chart as well. But today we'll be using the validation tool. There's going to be two windows that popped up. One is distance and the other is angle constraints validator. We're just going to ignore the angle constraints validator today. And then for our distance constraint validator, you can go ahead and sort these violations by their number of violations that they have. As you can see here, there is a lot of violations for ubiquitin. So usually when you open it, there is going to be rows that are gray. And these are not touched. These violations are enabled. So what I want to do is I'm going to disable these types of violations. So you can go ahead and click D, and then that row will turn from gray to red, as you see here. I'm going to disable this one again. And then I went ahead and disable any violations with a value of 20. And you can just go ahead and save. You can always re-enable them with E as well. So go ahead and save. Save, yes. Close it, and you've already saved it. So then the next step is to export our new violations to POKY Builder. I'm going to just save the new validations to our parent directory upon a Rosa trial, and finished. If you open up your main directory, you'll notice some new files called pros process. They can be saved as a UPL, a LOL, and a .aco. So back to our calculations, we just go ahead and exit out of these windows here. Already done using them. Now you can finally be using POKY Builder. So I'm going to set up my working directory as the main one. Just going to click open. Here it already auto-loaded my chemical shifts and my sequence, but in case it doesn't, you can go ahead and just click on this open sequence file. It can open using the sequence or the FASTA file. So I can just use, today I'll be using the sequence file. And then if it doesn't upload the chemical shifts, early in the tutorial, we did upload our chemical shifts via BMRB. So we just go and import it from POKY using the arrow from the far left side. Here is the chemical shifts. And now we'll be using our restraint. So there is a box called NMR through space data. This is where you can upload spectras, list files, peak files, but those files won't be available for us today. But if you have those data, you're more than welcome to use it. So we'll be using the restraints that we calculated. So the post process.upl file we're going to upload with the new validations. And then here you can go ahead and upload any information about your sulfide bonds, cis prolines, and then we can make sure our pack seed is checked to just to increase the accuracy of our protein. And since we are using our own restraints, then we'll be constraints to only X. But from the drop-down menu, you can go ahead and choose your different types of calculation. You can click pre-process, click start, and then once you click calculate, um, you will get another email similar that says that Ponderosa job has been submitted. Here it will give you a new job ID. And then once the calculation is complete, you'll get another email stating the job has succeeded along with the new job ID. As you see here, our job ID as in 4131. You can go ahead and download that zip file. I already went and unzipped it to our parent directory. Here it is. It'll basically show you the same information this time with the new validations. And you can go to best evaluated. Here you can see our new restraints and then our new structure as the form of final X or final.pdb. As you could see right here, final.pdb and then final x.pdb. Now we want to look what our structure looks like. You just go ahead and close POKY Builder. And then for the POKY app, you can also access PyMol. Just click on this button right here with the PyMol logo. Then the GUI for it will pop up. As you can see here. And now we just go ahead and upload our new structure. So we can go ahead and click open, go to our parent directory. Oops, sorry. It's gonna be users. My name is Karen. It is saved on my desktop. Ponderosa trial. The new job ID, which is 4131. And then best evaluated. So the difference between final PDB and final X.pdb is just the formatting of the PDB file. Final X just fits more for the XOR and IH calculation. So we'll just be using that. And here is our ubiquitin structure. If you want to assign the secondary structure to it, just click on this A button and do assign secondary structure. Now let's compare it to the PDB structure that's uploaded on the database. So we can just go ahead to fetch and we're doing the ubiquitin structure that is set by NMR and not the X-ray crystallization. So the PDB ID for that would be 1D3Z and enter. Here is the PDB database structure. You want to go ahead and align it. So click on the A again, scroll down to align to molecule final X. And now let's go ahead and center it. Here you can see that the structure is pretty close. Their almost D value, which shows the backbone alignment, has a value of 1.033, which is nearly perfect. You can, of course, increase accuracy by editing the constraints some more, getting a better nosy data, so on and so forth. But this is the tutorial on how to generate an accurate structure with simulated data with POKY Builder and with the help of Analyzer. Once again, my name is Karen, and thank you so much for watching.